Maddie Melson, and today I'll be presenting over imacloprid impairing memory and brain metabolism in honeybees. So for the background, we will first go over Apis mellifera and then insecticides. So what is Apis mellifera? Well, it's the scientific name for what's commonly known as the honeybee. And what do they do for the ecosystem? They pollinate many different types of plants and crops, and then they also produce honey. They are now all over the world because of their positive impact of the ecosystem as well as the economy. Because they pollinate so many different plants, it's very beneficial to have these, especially when growing crops. And while many different bee species also pollinate, Apis mellifera are seen as the main contributors to this. So insecticides. What is an insecticide? Well, they're chemicals designed to kill insects, and the specific one that we will be talking about today is imacloprid. How do they impact honeybees, you might ask? Well, imacloprid is a neonicotinoid pesticide primarily designed to control pests in agricultural settings, and it works by targeting the nervous system of insects. So these are they're commonly used in instances like farming when you don't want insects and pests getting into your crops. And then also, contamination can happen either directly or indirectly from residue. So directly meaning if a plant is or a crop is sprayed with this insecticide and a bee lands on it, they are now contaminated. But also if a crop brushes up or the wind blows and it can unintentionally get on a different crop where then a bee lands and now they're also contaminated. So for my research question, that is, does exposure to imacloprid affect memory and brain metabolism in honeybees? So for the article and study analysis, I found an article where the researchers administered oral doses of imacloprid to honeybees and assessed their olfactory learning capabilities using the proboscis extension reflex, or the PER paradigm. Olfaction is very important for honeybees and is often how they find plants and remember things and learn things. The PER paradigm is a classical conditioning method that researchers can use where they teach bees to extend their tongue when presented with sugar water. So for this specific experiment, they did this specific article, they did 3 different experiments. The first one studied retrieval, second one studied the short, medium, and long-term retention, and then the third studied the olfactory memory of the honeybees when exposed to this chemical. So for experiment 1, they tested conditioning and acquisition. Their objective was to assess the impact of imacloprid on acquisition phase and olfactory learning. Their method to do so was honeybees were orally administered imacloprid in specific doses, and which was dissolved in sucrose solution, so sugar water. And the bees underwent 1 trial conditioning using proboscis extension reflex, or the PER method, and then were exposed to an odor paired with a sucrose reward. For experiment 2, they tested the retrieval performance. The objective of this was to evaluate the effects of imacloprid on the retrieval phase of olfactory memory. They did this by conditioning bees, as in experiment 1, like we talked about, but imacloprid was administered 15 minutes or an hour after the conditioning trial, and the retrieval performance was tested 15 minutes after treatment. So this basically tested how well they were able to retrieve and remember the information when administered this chemical as compared to the control group. For experiment 3, they tested retention of memory, and the objective was to determine the impact of imacloprid on short, medium, and long-term memory retention. They did this by following one trial conditioning, bees were tested with imacloprid at various time intervals post-conditioning. So 30 seconds, 3 minutes, 15 minutes, 1 hour, or 24 hours. And then their memory retention was assessed 15 minutes after treatment. So this just tested how different bees would respond and be able to retain what they had learned during the PER method based on if they were given imacloprid or not. So the results found that imacloprid impaired medium-term memory when administered 15 minutes or 1 hour after conditioning, but not short or long-term memory. This suggests that imacloprid specifically disrupts medium-term consolidation. And then they also found that imacloprid administered during the retrieval phase did not impair bees' ability to retrieve the conditioned response, indicating that the pesticide affects memory consolidation rather than retrieval. So memory consolidation is how well they adapt and kind of hold on to new memories. And then memory retrieval is if they can grab on and recall prior information. The study also found that 30 minutes after oral treatment with imacloprid, the bees' olfactory learning performances were impaired, as evidenced by the reduced condition response during the PER procedure. Another study found that bees exposed to this chemical during their larval stage of development had impaired learning abilities in regards to olfaction and visual senses. So this basically shows that not only are bees who are working and out and pollinating different crops being impacted by this, but also if they're exposed during their development, they can be negatively impacted by this chemical. So with implications and significance, these findings indicate that long-term exposure to imacloprid could have detrimental effects to honeybee cognitive functions and potentially impacting their survival and ecological roles. This is due to it would reduce their ability to forage and remember where plants are, so they're not as effective in getting that pollen and nectar. And they also might experience an inability to navigate to and from the hive because they forget kind of where they're coming and where they're going. So here are my citations. Thank you very much.